We are finally at the end of this lesson, the last section of this lesson, which is part 5, on forces of evolution. So it's been a pretty long lesson, and we are looking at the last couple of forces of evolution, starting with genetic drift, which I defined at the end of the last portion, which was basically a change in gene frequencies due to a random event. And a lot of times this comes down to probability or chance. As we can see here in this table, we are looking at coin tosses, and we can see how the more coins you toss, the more accurate your results are going to be. We know that it's a mathematical fact that there's a 50-50 chance of coins being heads or tails. We can see that when we only toss 2 times, we are not getting the right ratio. We're getting a 2 to 0 chance, or a 4 to 6 chance, or an 11 to 14 chance. But as our sample size increases, once we get beyond 5,000 coin tosses, our odds of achieving the accurate ratio improve. So that's the important thing to remember with genetic drift, is that it can also be summarized as a sampling error. So something random has happened to wipe out part of the alleles, perhaps. In the slide before this, I showed you the man randomly stomping on the more green beetles than brown beetles, and in the next generation, the green beetles' proportion of the population was greatly reduced. So again, it's random. It had nothing to do with any environmental factors or any benefit or advantage or disadvantage. Those green beetles just happened to be squashed randomly for no reason, and it still resulted in evolution occurring because the gene frequencies changed over time. So here we are looking at different populations of different sizes, and how basically you can see the drift here, how genetic drift occurred in different size populations after only 38 generations. And we'll look at this potentially in the next couple of slides. Okay, here's another example. In a population called, or a religious sect in Pennsylvania called the Dunkers, they were a small population size, and their blood frequency, or their blood type frequency, is not representative of the greater population. So basically, if something big happens and wipes out a big portion of a population, genetic drift can occur. Or the same type of thing can occur when a small group is separated or migrates to a new area, and that is reproductively isolated. This is called the founder effect. So if a small population starts on an island, for example, is reproductively isolated, their gene frequencies are going to be different over time because of the small sample size. This has occurred, here's another example, when people migrated from Asia to North America and founded North America. They were a small groups, and their blood types were not proportionate, did not match up their frequencies of blood types with their ancestral population in Asia. So if a small group of individuals migrates to a new area and becomes reproductively isolated, again, that's called founder effect. So Native Americans descend from a small population of individuals that crossed the Bering Strait from Asia about 12,000 years ago, but while blood type 0 or O, blood type O, exists at a very high frequencies in Native American populations, it's relatively infrequent in East Asians. So what we're seeing is that whatever population ended up founding the population in America, the original groups of Native Americans that came over, they must have had more, randomly, just had more blood type O. And now it's greater represented in Native American populations than it is in Asian populations. That's another example of genetic drift. Okay, and I said all of this stuff already. Dun, da, 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 da. All right, as I said, while blood type O exists at very high frequencies in Native American populations, it remains relatively infrequent in East Asians. These data, contrasted with robust genetic evidence that Native Americans are most closely related to Northeast Asians, suggest that the small founder population of Native Americans that crossed the Bering Strait had by chance a higher frequency of blood type O than the parent population of East Asians. So, now we're moving on to another different mechanism of evolution, gene flow. So don't forget, genetic drift is caused by a sampling error, a small population size, or a random event. We're moving on to a different force of evolution now called gene flow. So as it says here in the bottom of this slide, for much of human history, and certainly today, genes are not isolated, they flow across populations. They flow across populations. So here, let's just review what we talked about earlier. We are going to look, you should be looking at the textbook for your information about the sickle cell trait to review, but don't forget a mutated version of a gene for hemoglobin results in the sickle cell allele. That allele can result in disease or adaptation depending on the genotype. An individual with 2 copies of that gene from a mother and father will have sickle cell anemia. An individual with 2 copies of the normal gene will have normal hemoglobin but no added resistance to malaria. An individual with 1 copy of each is heterozygous and has the biggest advantage in these populations. This results in what is known as a balanced polymorphism in which the heterozygous individuals are the most fit. So that was a quick review of what we learned in the natural selection segment about sickle cell trait, and now we are moving on to gene flow. Okay, gene flow. Gene flow is pretty easy to remember. It's the movement of genes from one population to another population. Gene flow keeps populations of a species on the same evolutionary lineage. For example, interbreeding of anatomically modern human populations and Neanderthal populations created gene flow. Think of pouring a new bucket of genes into a gene pool. This, of course, changes the gene frequencies in a population over time because you're getting genes from another population into your gene pool. Gene flow is also known as admixture. The basic idea here is that genes can flow across population boundaries, introducing new genetic variants into the gene pool. Here's an example with some humans of different hair colors. These 2 populations of dark-haired people and light-haired people mixed, and bada bing, bada boom, gene flow has occurred, and the next generation will have different gene frequencies. Another example of gene flow in human populations is the distribution of blood type B across Eurasia. So here's another blood type example. Our blood type O example was genetic drift. For some random reason, with the blood type O and genetic drift, the population that founded North America from Northeast Asia happened to have more of the O blood type than the ancestral population in Northeast Asia, resulting in a continent colonized with people who have higher frequencies of blood type O just due to that random sampling error. This example of gene flow is different. Here you will see the B blood type represented east to west. So we're seeing a greater proportion of blood type B in the east, and as we move west, less and less proportion of blood type B. So it's being introduced from east to west and flowing into these populations and changing by adding the blood type B from, again, east to west. So in our picture, notice that in East Asia, blood type B has a frequency of nearly 30%, but drops to nearly 0% in far Western Europe. The drop is not precipitous, it is quite gradual, also known as clinal, suggesting that nearby populations have exchanged genes and that the blood type B allele has slowly flowed east to west across Eurasia over the millennia. Gene flow can also be called genetic migration or interbreeding from one population of a species to another. This allows road-tested genes to flow into and out of populations. It also prevents populations from splitting into separate species because if the populations remain similar enough that they can still interbreed and produce fertile offspring, remember our concept of species at the beginning of this lesson, they cannot split up or become different species. So this mixture keeps a species together, it keeps the genes flowing, and I'll talk a little bit more about that as we near the end of the lesson here. This brings us all the way back around to speciation, which is what we talked about early on in the lesson. So, speciation is when a new species evolves. While microevolution refers to the changes in allele frequencies of populations, macroevolution focuses on speciation, the formation of a new species, and on evolutionary relationships among groups of species. Speciation can also be defined as the end of gene flow. So we're bringing this all the way back around and tying it into gene flow. Gene flow prevents speciation. If genes can still be swapped between populations, those populations are going to remain on the same evolutionary track. But once two breeding groups can no longer interbreed, for whatever reason, it could be behavioral, physiological, geographical, or any other factors, once they can no longer interbreed, they are no longer of the same species. There are other factors that we haven't talked about yet, known as isolating mechanisms, that can separate populations and lead to the appearance of new species. We did talk about geographical isolation. That can be an isolating mechanism, but genetic changes can cause isolating mechanisms as well. Since gene flow can no longer occur, the groups will become different species. So separation like this through branching is called cladogenesis, when one species gives rise to two or more descendant populations. Speciation can also happen without branching, as a single population accumulates sufficient new mutations over time to be considered a different species. This is called anagenesis. Because speciation is a process, it can occur at different rates. Scholars generally consider speciation by natural selection to occur at a very slow rate. For example, as a species slowly becomes better adapted to its environment over several generations. Sometimes, however, a genetic mutation, such as one involving a key regulatory gene, which is a gene that turns off or on other genes, can lead to the formation of a new body plan. Such genetic accidents may involve material that is broken off, transposed, or transferred from one chromosome to another. So that is one final example of how speciation can occur. This is the end of the lesson on forces of evolution. Make sure you can name and define the four forces, and make sure to review the sickle cell trait story and all of the factors involved. It is a complicated example. You can look to your book as well to kind of reinforce that information. And of course, message me if you have questions.